Hi there, welcome to Smith & Sheridan on Biotech, a podcast on the science and business of biotechnology, presented by me, Cormac Sheridan.
And me, Andy Smith. I can see you're well this week. How are you, Cormac?
I am quite good, actually. I have no major complaints to complain of.
Do you get my vein and attempt at a lead in there?
No, you're going to have to explain that to me.
I can see you're well, meaning we wanted to talk about ophthalmology this week.
Okay, boom, boom. The eyes have it.
Oh, yes, even better.
And when they don't, it's obviously a huge problem. And I mean, there's obviously multiple ways in which vision loss can arise. But I mean, there's two broad buckets, right, aren't there? There's kind of inherited genetic disorders, typically inherited retinal diseases that lead to almost complete vision loss over time, a kind of progressive vision loss. And I guess by the time you're middle-aged, maybe very little left of your hearing, of your vision, I beg your pardon. But then there's also the sort of diseases of aging, the macular degenerative conditions, which are much more highly prevalent, for which there have been some pretty nice biological therapies that have sort of really helped, I think, at least to stabilize the situation for many patients. We've had some limited success in the areas of gene therapy, a lot of failure, a lot of capital write-offs, because basically people invested vast sums of money in companies with little or no clinical data, and then later when they went into larger patient numbers, they just didn't get the kind of efficacy that they wanted. It's asking a lot, isn't it?
It is. It's strange, I mean, there's also another source of vision loss, which is injury, which you didn't mention, but there's very little of drugs you can do for that.
Yeah.
And even the age-related aspects extend to medical devices like cataracts and intraocular lens replacements. But we won't cover devices, we'll cover just drugs. And we have these discussions sometimes when we talk about the unmet need being so great, and it is in ophthalmology.
Yeah.
But as you're hinting, everything from photodynamic therapy to anti-VEGF therapy, we've come such a long way. There is so much progress that has been done for ophthalmic diseases that makes me, appreciative is the right word, in awe of the fact that this is an area where there has been active drug development and it seems to work.
Yeah, and has been, I haven't checked back the dates, but we are talking several decades of usage, aren't we?
20 odd years. And we have a discussion sometimes where we say sometimes when the first therapy is introduced, it ends up being the best. This is absolutely not the case in ophthalmology, particularly in age-related macular degeneration, which you were sort of hinting at. This is where you have blood vessels at the back of the eye as you get older. They leak, they swell, and you need something to stop that cell growth. And the first drug was not very good in terms of photodynamic therapy. It needed to be injected into the veins and then the laser to activate it at the back of the eye. And then we had what I'm sure is many people's idea of a nightmare, where you have a drug that's injected from the front of the eye with a syringe, and you can see it coming, if you're a patient, from the front of the eye to the back of the eye. So you've got hopefully a very skilled, they all are very skilled ophthalmologist, injecting a drug into the back of your eye at the retina. And it's got to be worth it, right? And it does seem to work. But we've had a whole progression of drugs that were anti-VEGF inhibitors from the Aptamer, from iTech, all the way through to Genentech, or Roche's now Lucentis, which have got better and better, both in terms of their target engagement, but also the thing you would want with an intraocular or intraretinal injection. The thing you would want would be to have fewer of them, right?
Yeah.
So you start off with monthly injections with many of them. And over time, the patient and the ophthalmologist agrees, and they measure their visual acuity, and they agree on a schedule for that patient. An ophthalmologist told me once is why very few clinical trials are actually done with existing patients, AMD patients who are already stable on their injection. They're almost all done with new patients, so you can establish that leading period, and then you can establish the frequency of injections after that to keep acuity stable.
But that's a kind of a no-brainer, if you like, though, isn't it? Because, I mean, if you have stabilized somebody, you don't want to go mucking that up. I mean, if you have some sort of a condition that is untreated going to lead to vision loss, you don't want to sort of in any way want to upset the sort of apple cart at all there, do you?
And no, and it's fortunate for drug companies and less fortunate for patients. But it's fortunate for drug companies that this is a very big market.
Yeah.
Age-related macular degeneration and the conditions that lead on to it.
Yeah.
Geographic atrophy, diabetic retinal retinopathy. Thank you. So those are very large markets. So as we're going to talk about, I'm sure, with gene therapies, the risk is that if you have a one and done therapy for a particular indication, if it's an inherited indication, like the first approved gene therapy, Roche's drug, Leber congenital something else,
amaurosis. Yeah. But you can also call it biallelic RPE65 mutation-associated retinal dystrophy.
Yeah, if you'll excuse me, I'll try not to mention it again, so I won't have to mention it. But if you develop a drug for that sort of indication, as Roche has found out with spinal muscular atrophy, their gene therapy, you pretty quickly, particularly if you have prenatal or postnatal testing, you pretty quickly treat the worldwide population, and you're relying on an incident population of a very rare disease, which is not a commercially viable market.
I don't even think, though, that Roche or Spark Therapeutics, and I think Pfizer was on board as well at one point as commercialization partner. Yeah. I don't think they've saturated the market. They've never reported sales data.
I think they do. They report, it's about 1 million Swiss francs, I think, last quarter.
Okay, well, that sounds like about one patient.
Yeah, probably.
I didn't think they were reporting it, but they also did some write-offs as well, because Roche paid 4.8 billion to acquire Spark, and I don't think that's been repaid in terms of revenue. Now, obviously, you can buy companies for other reasons, expertise, etc., but I don't think. I mean, I was trying to get my head around why it's a commercial failure. And it just seems to me that there's just kind of a very broad variation in the level of patient response, which is not surprising.
Yeah, that's true. But we don't seem to be finding that with the latest, not just gene therapies, but CRISPR's now is rearing its ugly head in ophthalmic diseases.
Yeah, it is. And it's kind of interesting that there was a sort of a ripple of publicity earlier this month, because Editas Medicine, at least their clinical collaborators, reported the phase 1/2 data for EDIT 101, which was the CRISPR-Cas9 gene editing therapy for CEP290 associated retinal degeneration, also known formally as Leber congenital amaurosis, by the way. And basically, that's CEP290 is the centrosomal protein required for photoreceptor function. And many of those in the US, at least according to a paper I read, 77% of the cases in the US arise from pathogenic variant IVS26, which disrupts normal expression of this EP290 gene by including a cryptic exon in the mRNA, which leads to a dysfunctional protein. So they were just correcting the mutation, and it was the first ever clinical trial of an in vivo CRISPR-Cas9 based therapy. So it has a certain kind of, it goes back quite a number of years that this thing got going. And it's interesting that the clinical trial investigators in the New England Journal of Medicine, May 6th, for those who want to check out the reference, they said of the 12 adults and 2 children that were treated, and they got different levels of dose, low, intermediate, and high.
Yeah.
So 6 had a meaningful improvement from baseline in cone-mediated vision, and cone photoreceptors are responsible for sort of sharpness and color definition, sort of bright light vision, I suppose. 9 had improvement from baseline in best corrected visual acuity. 6 had a meaningful improvement from baseline in vision-related quality of life scores. But Editas, back in November 2022, disclosed that only 3 of the 14 patients met its criteria for a positive response, and therefore they decided to pause enrollment and look for a partner, and I think the whole thing is stalled, and it's not really mentioned on the Editas pipeline chart anymore, so it's kind of shelved.
But in that New England Journal paper you mentioned, was there a dose response? Because it's strange, I think the jury's still out there on whether gene therapy will have a dose response. I'm sure if you treat more cells in the liver, then you've got less to turn over. But in other cells like the eye, that might not be the case.
I don't recall them breaking it out.
The reason why I asked, Cormac, is because in more recent announcement by a company, Resolute, isn't it, which is looking at an oral drug, I skimmed the announcement and it said they had sort of a flattened dose response. at the 200 mg dose. I couldn't tell whether that was the mid dose or the high dose or the low dose. I take it wasn't a low dose because they said they're going to stop at 200, which is unusual, I think, for an oral drug. But for a gene therapy, you'd expect more of a one and done effect, or one dose and done effect, rather than 2 or 3 doses.
I think there's so many moving parts, especially a first generation CRISPR-Cas9 therapy, that I think you can see a dose response when you've got the PK, PD stuff down. And here there's so many imponderables around delivery and also just the manufacturing process as well.
Because you inject a few microliters into the back of the eye. You would have thought, what's the difference between 1 or 2 microliters? In a small organ like the eye, you'd think there wouldn't be much of a difference, but you never know.
Yeah, and I haven't sort of seen any reference to a dose response in the paper. As far as I know, I mean, obviously the main objective of this thing anyway was safety.
And at least they've got positive responses, which is great news for patients. But going back a bit, when we talk about the anti-VEGF drugs for AMD, as you've mentioned, there have been graveyard drugs, there have been moderate improvements, partly in terms of efficacy, but also in terms of reducing the frequency of injections. But we've also been through the phase where we've had a number of companies trying add-ons to anti-VEGF therapy. And if it's one thing that puts me off having one injection into your eye, it's having 2 at the same time of 2 different things. And that's what put me off by a company called Ophthalmic, which had a clinical trial, well, actually it had 3 clinical trials to add on to their, I think it was platelet-derived growth factor inhibitor to an anti-VEGF. So 2 injections to the back of the eye.
What's that company called again?
Ophthalmic.
Oh, yes.
It failed 3 phase 3 studies. I think it had a deal with Novartis. It was the same management team that brought you the disaster known as iTech with their drug. But for listeners who might want, one of my learnings from being an investor for 20 odd years is when a company has an experimental drug and they call it by a brand name, right? So they go to the trouble of registering the brand name while it's still in phase 2. That is jumping the gun a bit. And this is what Ophthalmic did. They called their drug, their PDEG inhibitor, Fovista. So they registered the trade name. And that always worries me, right? Because are they getting a bit ahead of themselves? And unfortunately, they did. But they're not the only company to, as you mentioned, fall by the wayside over the years. There was a Belgium company that had a drug, ochriplasmin, I think it was called, that actually got approved in the end. Do you remember that?
It got approval, right, And that was a company founded by Desiree Collin, who had a big success with an antithrombotic agent.
Yes.
And the same principle was applying here. It was sort of
that's right. So a different sort of ophthalmic indication. So, you get things floating around in the eye, or you get damage to the back of the eye. Yeah. And this enzyme apparently breaks, and the product was Jetria.
Or ochriplasmin was the Thrombogenics was the company. And
that's right. They then changed their name, but then had
they were a company who were riding very high, that they were sort of a classy company that was well managed and raised lots of cash. But I think they readily admitted that maybe it was a very complicated condition that they were trying to treat, macular adhesion.
Yes.
It was very hard to sort of access the patient population. I think that they sort of maybe didn't fully understand the market. They'd done a good job of getting it through the clinical trial and regulatory review process.
Yeah, and I think it was eventually withdrawn from the market.
I think so.
So, I mean...
And I think as well, surgery is maybe a better bet than they thought it was.
Yes, the surgery to the back of the eye, another thing that scares me.
But I've been looking at it, I suppose, the lens, if I can use that term.
Oh, yes, another one.
Which I've been looking at a lot of ophthalmic diseases has been the genetic lens, because that's where, say, there is a lot of unmet need. But it's also, I think that in particular with the sort of classical gene replacement approaches, the eye is one area where you find yourself in a blind alley. I'm sorry for using that term. I know, a bit inappropriate. But the big problem, though, is that you have a plethora of different genes associated with the pathology. And within those genes, we've over 20 inherited retinal disorders to begin with. More than 260 genes associated at the last count. This was something a piece of work I did a little while back. And then within each of those genes, there will be hundreds of disease-causing mutations quite typically. So that you're looking at thousands. So you can't do gene editing. easily. You may be able to do gene replacement, but you're not going to do 260 gene replacement therapies for these markets. There's no way of doing it unless at some future point where we have the manufacturing piece down and there's some very clever way whereby you can just swap in or out a gene cassette into an existing infrastructure.
No,
but
absolutely.
Gene therapy is not at that stage yet.
It's not
really
yet,
isn't.
But there are hints. I mean, you're not using your imagination, Cormac. I mean, we've talked...
I admit that readily. If I had more of an imagination, I might be a successful biotech entrepreneur.
But we've talked in other podcasts about how cancer therapy with gene editing is getting to the stage where it's the algorithm that designs the cassette that inhibits certain genes in some patients and not in others. This is what you're talking about, right? But so the algorithm may be one day that programs the gene editors or the gene editing units that then bind to the genes. The algorithm may be the thing that's approved by the FDA.
But see, the one thing I used to be very careful about here is that with cancer, you're trying to kill something that you don't want.
Yeah.
Whereas with ophthalmology, you're trying to
correct something that you do want, possibly.
Or preserve remaining function or delay deterioration. So you have precious, because very often you can't go in too late, because obviously if the retina is completely destroyed, there's nothing to preserve anymore or to build on. You have to have functioning photoreceptor cells before you can put in anything. So you do have to have some level of vision, and you sure as hell don't want to start doing anything to harm that. And I know these gene therapies and the procedures around them, they do carry risks, and the safety statements that developers set out say for like extensive and sobering reading for anybody who's maybe putting their kids through the procedure. And it is a risk, and obviously there are rewards, but it's not like a cure. And even those who have a good response, we don't know yet still how long that response will last for.
And that seems to be the common factor for all for the ophthalmic diseases drug development that we've been talking about. As AMD and geographic atrophy, you might just be stopping progression. So you're stopping vision loss.
And that's okay. I mean, if somebody is diagnosed at the age of 75 or 80, okay, you'd rather not have it. But if you can, say, maintain vision so that, say, they can still drive, they're drivers, and they can obviously still conduct the kind of activities of daily living without any problems. I mean, that's a big positive intervention to making somebody's life.
We can both agree on that. But I have read somewhere where, someone's talking about, well, this is a quality of life improvement, right? To see more effectively, to see more lines on a vision chart, or to be able to see faces rather than just shadows. It's a quality of life improvement. And why isn't money being spent on curing cancer and making people live longer? Well, vision is pretty bloody important, isn't it? I don't agree with the quality of life thing. I think,
I totally agree. I mean, you could say getting a yacht is a quality of life issue, but it's not the same thing. And I would reject that type of argument wholesale. Obviously, you treat people with cancer. Obviously, you preserve life, but obviously, you also preserve vision. Obviously, you preserve, if you can, hearing. They shouldn't be put in opposition to each other. They're all important. But just to get back to the gene therapy stuff, though, what was interesting to me was that there are approaches underway that are trying to get out of those individual single gene silos and look at more broadly functioning gene therapies that are kind of mutation agnostic. So, say, sparing vision in France was an early kind of player in this field, and they are now in a phase 1/2 trial with their lead program SPVN06. So that's an AAV-based gene therapy expressing two versions of the same protein. It's called rod-derived clone viability factor. Now, there's almost kind of the naming of the protein itself almost has a sort of a marketing kind of dimension to it, doesn't it? And it has a related isoform, rod-derived clone viability factor long form. So that the long isoform reduces oxidative stress, so that's some sort of enzymatic activity. And the regular isoform, it provides some sort of metabolic support to the cones, as far as I know. Because what's very interesting about, say, RP especially, is that most of the mutations in RP arise in genes that are expressed in the rods, which are the photoreceptors that sense light in low light conditions. When the rods start to deteriorate, there's a sort of a bystander effect on the cones. And so it's a kind of a two-step process, so that in some way, I think either that the rods provide some sort of metabolic support to the cones, or else that when those rods are no longer available to absorb light, that the cones then are damaged by exposure to excessive light. And also just the destruction of the rods can just create a sort of a toxic microenvironment within the retina. Yeah. And that may also destabilize the whole situation. So sparing vision's idea is that they want to hopefully arrest or at least slow down the rate of vision loss. And I think they'll have data to report on that, I presume, in the not too distant future. And that's going to be interesting, because that is something that potentially could be aimed at a fairly large number of patients. And actually, even before that, Ocugen, Malvern PA company or Pennsylvania, they have a similar idea that they're, again, mutation agnostic. They have an AAV5 vector expressing NR2E3. It targets nuclear hormone receptors, modulates gene expression within the retinal cells, and the idea is that it sort of resets retinal homeostasis and stabilizes cells and rescues photoreceptors from any further degeneration. So they've reported phase 1/2 data, and it's a small trial, 83%, but only 10 of 12 patients had stabilization or improvements in their treated eyes in terms of best corrected visual accuracy scores. And various kind of percentages had 4-letter improvements on eye charts and 7-letter improvements. 42% had a 4-letter improvement, 33%, 1/3 had a 7-letter improvement, etc. So it's not, again, slam dunk, and it'll be very interesting to see what data they come up in the phase 3.
But I do like the sparing vision ethos, if you like.
Yes.
That vision loss in a wider population is a one-way street. And I think back to optometrists, high street optometrists, or at least consultant optometrists, will talk about catching vision correction early. Yes. Because it then preserves the vision for later life. If you go for years without having glasses or contact lenses, then by the time you get it diagnosed, you've already lost a lot of flexibility in the eyes and that sort of thing, and the lenses and things. So I absolutely agree with that sparing vision ethos, because it seems to run through even the low-tech side of ophthalmology. Yeah,
and Ocugen have the same ethos, because it's the same, they're mutation agnostic. I mean, actually, it's interesting that J&J are still in here, and they have a mutation-specific gene therapy. And it's very interesting, though, that their capital outlay is a fraction of what Roche paid to pick up Spark. J&J acquired this program from their UK partner, Myra GTX, just last December. And they paid $130 million upfront. Sorry, that's upfront and near-term milestones. So much of all been paid.
Change for J&J, isn't it?
And they'll pay another $285 million on first commercial sale, and on when the technology has been transferred. This is for voretigene sparoparvovec. Apologies to everybody concerned there. And that's expressing an isoform of a protein called ORPGR, which is mutated in GPR associated retinitis pigmentosa. And confusingly, ORPGR stands for retinitis pigmentosa GTPase regulator. But obviously mutations, and it's an X-linked condition apparently, so it affects boys. But it's a truncated gene cassette they're putting in, which apparently improves the stability of the resulting protein, but doesn't affect its actual localization or functioning. It's just they've knocked out a kind of a purine-rich region, but they still get the enzymatic activity. And that rescues the rods and cones that are affected by the mutation. So that's in phase 3. And it's just interesting to see that there is still some big pharma presence there, but you sense there's still innovation. And I do want to mention one company that I find completely fascinating in terms of their approach, because it's not mutation agnostic, but it does manage to address multiple mutations with one therapy. And the way they do it is so clever. This is a company called Ascidian Therapeutics, and they're in the news in the sense that I think their idea has been accepted by the FDA, or else they filed it, and I think they're due to commence a phase 1/2 trial imminently. And Ascidian are doing something that nobody else that I'm aware of is doing. They're introducing to human cells something that doesn't ever occur in human cells.
Yeah.
They're doing trans splicing, And they're called ascidian because I think there's certain marine invertebrates, sea squirts, are a more colloquial term for them, which do this. And basically it means, because we do cis splicing, so that when we're processing our pre-mRNA to mature mRNA before translation, we knock out the introns and we splice together the exons, but it's all from one molecule on the spliceosome. Whereas you can also do trans-splicing, so you can take bits of exons from one pre-mRNA molecule and bits of exons from another pre-mRNA molecule and bung them together. And so it's a very, very clever way of trying to get around whole swathes of stretches of the gene. And the one that their lead program is ABCA4, which is this massive big gene and massive big protein, multiple mutations. And by knocking out huge swathes of the regions of the pre-mRNA that causes disease, they can address with one therapy 60% of Stargardt disease causes, which is a certain inherited retinopathy, and they're going for other retinopathies as well. And I just think that's so innovative, and I think it's very clever. And they deliver the RNA exon editing components in an AAV vector. It's encoded in DNA, and it's like an RNA molecule that just seems to sort of provide all the bells and whistles needed to achieve this trans-splicing effect. And it's just incredibly clever, and it's going to be very interesting to see whether it actually works or not.
And is this the mechanism of action you described in, I think, your Nature article, wasn't it? And I read in there that, isn't it, fish and other amphibians are able to repair their eyes,
yes.
But we humans are far too complicated and proud to be able to do that.
Yeah, that is exactly true. I mean, fish, reptile and bird species, they can regenerate retinal tissue, and we can't. And that's why we have all these problems. And there are efforts underway. There's various people trying to sort of do this kind of idea of reprogramming. And it's really early stage. There are some people who are claiming to make progress, and say there's an interesting company in Switzerland, Endogena Therapeutics, and they claim that they can do this with small molecule inhibitors of kinase mediators of the Wnt pathway. They claim that they can reactivate retinal stem and progenitor cells in the ciliary epithelium, and these migrate to the retina, mature into functional photoreceptors. And it's a big claim, and quite a few other experts think that you can't really achieve that with small molecules. You really have to go with transcription factors. So,
but people will say, why are we mucking around with either inherited retinal diseases with tiny numbers of patients, or even some of the bigger specific subsets of RP retinitis pigmentosa? Because it's not enough patients. Why aren't we going straight to these cutting edge things you're talking about, where you restore vision where they're not there? But in the same way as we have cancer therapy, right? That starts off with the sickest patients first, because they are less risk averse, and you might be at a later stage, and you might be able to show an effect in those patients. And that's where we are today, even with gene therapy and gene editing in ophthalmic diseases.
Yeah.
But even in those young patients who actually can see things they couldn't before, I've used the phrase, we're bordering on biblical, aren't we? Where they couldn't see something before, and now they can. And this should be a very encouraging step for what comes after the things you've just been talking about.
Yeah, I agree. I think that, say, some of the recent clinical successes in restoring or even allowing hearing to take place in a baby who never heard before, that kind of stuff is arguably sort of not only bordering on, but in the realm of the biblical. And we've had sort of modest clinical successes, but they're not kind of in the area of ophthalmology. I don't think we've really had that sort of, it's a miracle moment yet, or a eureka moment. And as I was saying earlier, it's a huge amount to ask, because there's so much metabolism that happens in the eye. There's so much energy consumed to achieve what in itself I find miraculous, that idea of phototransduction, that light energy generates ultimately a chemical and an electrical signal that
we can then interpret, the brain interprets.
Yeah, but I mean, it's amazing. And so much can go wrong there. That's why we have so many different types of vision loss indications.
Yeah.
And I think what the field is doing, to its credit, is that it is learning how to deliver proteins.
Yeah.
It is learning what does or doesn't work, or works in a sort of an intermediate fashion. I mean, I did look at a video, totally promotional video, by Roche about the experience of one family with two little girls, both of whom have the biallelic RPE65 associated corneal dystrophy, and they both underwent the procedure about a year ago. And it's worth watching. It's very sweet and very moving, and it's not the full clinical situation, but it's anecdotal. But going from one positive anecdote, and obviously you wish them well, but they're only 1 year out from therapy and there's a long way to go. But going from the anecdotal to large-scale trials and, importantly, real-world data as well, real-world studies, where you can see these operating in patient populations over long timescales. It's early days, and at the same time, I really do salute the people involved. It's a very heavy lift, and there is progress being made. And I think, even just sort of interesting enough, there's a small German company called Diabetic, and instead of doing subretinal injection of their vectors, they're doing intravitreal injection, which apparently is far less invasive. So again, they're going after just a single gene-associated retinitis pigmentosa. In this case, it's the CNGA1. It's a subchannel. I think it was a subchannel of an ion channel that's found in the rod photoreceptors. So they've just gone into the clinic lately. But their vector technology is done through in vivo screening in animals, I think, and they pick out ones that are just very adept at getting into photoreceptors. So that's an interesting technology. Even if this particular clinical program isn't successful, that might be an interesting technology as well, if others were able to get their hands on it, if it works.
And as an interesting aside, one of the interesting aspects of clinical studies in these sort of patients is that you have a patient with two eyes, right? They both have the same mutation. You have effectively a control. And
you do, yeah.
Normally the one eye gets treated in a clinical study. Can you imagine the frustration if you're one of those patients? Look, I can start to see something in one eye. When are you going to treat my other eye? And the FDA will say, well, wait 15 years to see whether that one's okay, and we'll treat the other one.
With Luxturna now, though, both eyes are being treated, but there is a
really?
kind of...
but that's outside the clinical trial.
But they're not treated simultaneously. There is a gap, and that's obviously sensible and intelligent. But
well, that's going to confuse the parts of the brain that deal with visual acuity, isn't it?
Yeah, well, I guess you can wear a patch over the treated eye at one point, and then maybe wear a patch over the... But I know that the eyes, they have this kind of... Vision itself has this kind of adaptive plasticity.
Yeah.
So it's been one of those areas. It sparked a lot of excitement, and I do think that maybe the Luxturna story was slightly overhyped in the sense that when you break it down in terms of what actually got them their actual approval, I mean, it was their ability to navigate a certain route in levels of low light. And it's a very complicated scoring mechanism, but there was a definite difference versus the placebo arm. But that's what it was. It wasn't a sort of a slam dunk. But at the same time, you often say that the first drug in a certain indication is often the best one. I don't think that applies at all with gene therapy. I think it might apply with something like statins or something, even though there it kind of didn't either. I do believe that, incremental innovation is very important in pharma and in biotech. And even if you get the basics right and the biology right first time, there's always ways of just kind of finessing what you have, even if it's just to say reduce the frequency of injections if you're talking about a biologic. I mean, that's very meaningful. And
I agree. We're still at the stepwise thing. So the clinical endpoints that was established by Spark with the low light maze that patients have to go through, that's established as an endpoint, as well as the visual acuity aspects for the wider thing. So it's baby steps.
But at the same time, I suppose what's discouraging, and sorry to introduce a downer of a note, but like obviously Roche has had to do a big write-off on the Spark acquisition. Biogen and Novartis have also been burnt. Biogen put down 800 million or so, I think, to get Nightstar Therapeutics.
Oh, yes.
And they had two late-stage failures, one in X-linked RP and one in choroideremia. They paid the money in 2019. They did the deal. 2 years later, a phase 3 and a phase 2/3 trial both failed within a month of each other, actually. So, I mean, kudos to Biogen. They
trying,
They put the money down and they took the hit, but it got done. And the same with Novartis. They acquired Gyroscope Therapeutics for big money, 800 upfront and 700 million in milestones that I'm assuming never accrued. And again, there was a late-stage failure there. They were doing GT005. And this was interesting, though. They were going for geographic atrophy secondary to acute macular degeneration. So that was a prevalent indication. It was one of the few gene therapies for prevalent indications that I've seen. Yeah. So it could have been huge for them. And it wasn't. September last year, they terminated that program. But they were trying to put complement, basically, some sort of a complement inhibitor into the eye because it was excessive complement activation, they believed was one contributor to the progression of the condition. So those are kind of negatives, and you'd imagine that they won't be biting anytime soon. And at the same time, obviously, a flow of dollars is needed to maintain the sort of momentum in terms of the clinic. So the field does need success, continued to secure the continued investment. And Beacon Therapeutics is in there. They're doing, again, X-linked RP caused by mutations. It's actually the same thing, actually, as, or similar rather, to the J&J program. It's also focused on the GPase regulator. So yeah, Occlusion's phase 3 could be very important in that respect. And the market doesn't love them. I mean, their market cap is modest, but that's an interesting program and it could be important. But
well, the market didn't love Resolute until it announced its phase 2 study.
Yeah, and it kind of loves them now. And I'm sure the early investors in Resolute must love them even more.
Yeah.
Yeah, it'll be interesting to see how that plays out, because to me, it looks like they're not being taken very seriously. And it's like a small molecule that is addressing indications that currently require injections in the eye, right? That's the basic idea.
Yeah, and in the same way, AMD, and the bar is pretty low, right? So the alternative is to have an injection through the front of your eye to the back of the eye, or take this tablet, or even at worst, or not worst, but at approaching worst case, take fewer injections and take one of these tablets once a day. That's positive. So I think the market for Resolute is waiting to see who they partner with, because anyone that does an anti-VEGF therapy at the moment is potentially a partner, and that ties the clinical trials in to their anti-VEGF therapy and Resolute's drug.
But I suppose I guess people are waiting because they've reported some sort of their endpoints. Yeah, a reduction in central subfield thickness.
Yeah, and again, I mean, when I read that, as you know, I thought it was a bit. It's a surrogate marker. I mean, I know it's probably almost certainly measured by OCT, which is laser scanning of either the front or the back of the eye, depending whether it's an anterior or posterior scan. And that's real structural changes to the eye, but it's not the thing we normally want for the visual acuity, the letters on a chart or things like that. But it's early days, and I think the market is worried that Resolute don't have the money to go into phase 3 or the partner to go into phase 3 and measure those clinical endpoints. But we just have to wait and see. Hopefully someone will come up with the money to do it, and we will get to know whether it works or not.
Yeah. Okay. So we're at this is the sort of macabre stage of a company's development, hoping that something will turn up. But as ever, data drives deals and data drives dollars. And so I guess it's the data that we're going to be seeing from a couple of key trials is going to determine what happens next in this field.
It does, as always.
And fingers crossed, it'll be good data, because who can argue against something that is going to restore or maintain somebody's vision?
Quite.
Thanks for listening, folks. Until the next time, Andy, see you.
See you next time, Cormac.